All right. Good afternoon again, everyone. So once again, my name is Jennifer Shera, and I'm part of the community education staff here at California Botanic Garden. And I welcome you all to today's program, Seed Biology for the Seed Collector, which is going to be led by Cheryl Birker. We're going to get started with the program in just a minute, but before we do that, I wanted to go over a couple of things that will hopefully make your webinar experience go just a little bit better. So just in case you need to leave today's program early today, or if you want to share this webinar with someone else, or if you want to be able to refer back to something at a later time, we are going to be recording and uploading the presentation to our CalBG YouTube channel within the next couple of days. So go ahead and keep an eye out for that. During the webinar today, we are happy to receive your questions, but we are going to be answering them during a Q&A time at the end of the program. So please go ahead and type those into the chat or into the Q&A box, but expect that we're going to answer them at the end of the program. So you can have them all waiting there, all queued up. We also want to extend a thank you to the National Park Service and the Forest Service for their support of this webinar today. And I am going to go ahead and turn things over to Cheryl now to get things started.
Thank you, Jennifer. So hi, everyone. I'm Cheryl Birker, and I'm the Seed Program Manager here at California Botanic Garden. Let's get this started. So Jennifer already mentioned some of these things, but just, welcome everyone. Really want to make sure to extend our thank you to the National Park Service for funding this webinar, with special thanks to Steve Buckley for support and assistance. So National Park Service has contracted CalBG to host a series of trainings. So this is not the first webinar. There was also a webinar back in 2023 on seed processing and storage, sort of just like post-collection care of your seed collections. That is posted on the CalBG YouTube page. We also did a webinar in 2024 on seed collections in restoration practice, reviewing seed transfer zones and seed bulking methods. So both of those can be viewed on the CalBG YouTube page at any time. And then this presentation will also be up in a few days. And we will have more seed trainings coming in 2026 as well. And then as Jennifer mentioned, go ahead and put questions in the Q&A and we will address those at the end. So a little background on me. So I'm the manager of the seed conservation program at the garden. This is the California Seed Bank, the largest seed bank dedicated to conserving California's native flora. But fun fact, I actually got started as a Seeds of Success collector at the garden back in 2014. So I've been a seed collector for over a decade, and we have lots of experience with the Seeds of Success program. I know sort of the target audience for this webinar is collecting teams, making collections for Seeds of Success. But the seed bank that I manage is a little bit more focused on rare plants of California and long-term plant conservation. So trying to get seeds backed up in storage in these freezers before something happens to them in the wild. And it's sort of a safety net for species extinction. Because the idea is that if you process and store seeds correctly, many species have the ability to stay alive in storage for a very long time. In fact, we've seen this amazing seed longevity even in the wild. So back in the 60s, some researchers found seeds of this Judean date palm during an excavation of this mountainside fortress in near the Dead Sea. And radiocarbon dating estimated the seeds were 2,000 years old. And in 2005, scientists were able to germinate those seeds. So now there's a living plant that was grown from 2,000-year-old seeds. So we know that seeds have the ability to live for a very long time under the right conditions. And that makes seed banking a really cost-effective means of conserving plant diversity, right? We can go out and make these collections in the wild, bring them back to our facilities, process and store them, and they can be available for us in the future for safeguarding species from extinction, for using them for reintroducing species that are going extinct. We can use seed banking to conserve material for habitat restoration and also conserve material for future research. So we know that, seeds have the ability to stay alive long term. And you might sort of ask the question, well, how long can seeds live in storage? And the answer is that it's species specific, right? Some species are quite long-lived, others not so much. And so this graph over here, let me just move my thing. This graph over here is pulled from this publication, Long-Term Storage and Longevity of Orthodox Seeds, a Systematic Review. And so they were actually looking at, this paper is a meta-analysis looking at 6 different publications that were looking at many species and looking at seed longevity. And so on the x-axis is the 6 different publications they were looking at. You can see that a lot of them were working with crop species. There is quite a bit of research on agricultural species. And seed biology, as you can imagine, right? There's sort of a lot of funding in agriculture, but there are publications in here that we're looking at wild species. So this is Christina Walters from National Laboratory for Genetic Resources Preservation. You'll hear me mention Chris Walters a couple times in my talk. I'm sort of a fan. But this study was looking at 276 wild species. And then the y-axis here is P50, which is the time it takes for seed viability to decrease by 50%. So it's just a term that we use when we're talking about seed longevity and the lifespan of seeds in storage. And so from this graph, you can sort of see that there's quite a range, right? Some species, the P50, right, the amount of time it takes for half the seeds to die was over 800 years. Others, it was less than 100 years, but it's really quite variable. But I wanted to point out that the other thing this paper was looking at different sort of storage methods, how we're storing those seeds long term. And so here on the code for each of these studies, the ones that have AM before them are seeds that were in ambient storage. So ambient temperature, ambient humidity. And then the ones with CO were in cold storage. So you can see right from this graph that really the storage, how we're storing our seeds greatly impacts how long they are able to live. And seeds cannot live as long in ambient conditions as they can when they're dried and stored in cold conditions. And so I'm going to mention that a couple different times in my talk. There's also some interesting research on specifically California native plants. This Went-Munn seed longevity study was initiated in 1947. And they chose 91 different California native plant taxa and stored them in these glass vials. You can sort of see them here. These are all screenshots of this Life magazine article, which is sort of fun. It's like from 1948. An experiment that may last 360 years. Project will give botanists new information about the heredity of plants and the lifespan of seeds. And so they set up this experiment in these glass vials, different California native species, and these are under really low humidity, but the vials themselves are kept at ambient temperature. So that was thought to be like the most high-tech thing at the time, right back in the 40s, was this is how they were trying to store seeds to keep them alive long term. And they're set up so that germination testing is supposed to occur roughly every 20 years with follow-up testing until 2307. And this is a graph provided by Chris Walters at NLJRP. They have the seed samples at the National Lab, in Chris's lab. And these are data averaged for 70 species showing more than 0 germination. So for the species that are alive, right, this graph shows that the seeds are holding up relatively well. And then this graph also shows this sort of side experiment that's being done where periodically when they remove seeds from the glass vial, they will keep those seeds at ambient humidity. So instead of keeping them dry, they keep them at ambient humidity and then run these follow-up tests. And both times they have almost immediately died. So again, sort of another reason why it's very important how we're storing our seeds in order to keep them alive so that they can be used in the future for their intended purpose. And so not all species are long-lived. They're sort of different species have different storage behavior. So we have these three categories. There's orthodox seeds. Those are seeds that are known to be long-lived in storage. So things like Clarkia, Astragalus, Dudleya can live a very, very, very long time, and that is known. Then there's intermediate species. These seeds are known to be short-lived in storage. They might only live like 3 years, 3 to 5 years. So we know that salix, right, willows are short-lived. Some pinus are short-lived. Several species in Campanulaceae, particularly in Hawaii. And in general, there's a higher portion of species that are short-lived in more tropical climates. And then there's recalcitrant species. These are species that aren't storage tolerant at all, and they have a very short shelf life. So they will die fairly quickly. So things like oaks, walnuts, buckeyes, they actually aren't able to be dried. So the exact sort of reason why we can't dry them has to do with their cellular dry mass. So they're more fatty, right? Imagine like an acorn or a walnut, right? Have like pretty fatty seeds. So because of that high lipid content, they don't have very much cellular dry mass. So when you dry them down, they're damaged and it kills the cells and it hurts the seeds. So that's why we can't store recalcitrant species. But intermediate and recalcitrant seeds usually aren't good candidates for seed collection unless they're going to be used right away for restoration or reintroduction, right? So when we're making collections for the Seed Success Program, where there's, some future restoration that the seeds will be used for, we want to be collecting seeds that will survive in storage for some length of time, right? We don't want to be collecting things that are just going to be dying because they are recalcitrant or intermediate. But there's also this broader term for species that aren't very good candidates for seed collection. And we call them exceptional species. So within exceptional species, there's seeds that are recalcitrant, seeds that are intermediate, right? Short-lived seeds. But there's also these other categories. So some species are considered exceptional because they don't produce seed or produce very few seeds. Sometimes they produce seeds just fine, but they are low quality or inviable seeds. And we have some taxa on our Seeds of Success target list that I would sort of put into these categories. And we keep them on our target list because we keep looking out for them, right? We keep holding out hope. We still check for seeds. Maybe, we just need a really good rain year or something, and they'll finally produce some viable seeds. But we aren't hedging our bets on them, essentially, because I would consider them to be exceptional species. And then this last category, sometimes we consider something to be exceptional when seed propagation is just very, very difficult. And it is like impossible to get living plants from those seeds, right? Sometimes when we're propagating plants, for species that don't really propagate by seed, we have to take vegetative cuttings or divisions, some other means of propagation other than seed. And so if you're, collecting for a restoration project and there's a species where nobody knows how to produce plants from that seed, that might not be the best target for your seed collections, right? Your efforts might be better spent elsewhere until we can sort of figure out the proper propagation protocol from seed. And so, in general, more research is absolutely needed on seed propagation, on seed longevity and storage behavior, right? There's a lot of taxa where we don't know how long-lived they are in storage. We don't know whether they can be dried. There's also a lot of species where we don't know how to propagate them from seed, or we don't know how to amplify the seed during like seed bulking. And so there's a lot of really great research going on out there. We're participating in a bit of it at the garden. We had this seed amplification experiment that we conducted for the Bureau of Land Management, the Mojave Desert Native Plant Program in 2022. And we hope to get more funding to continue that work in 2026. Trying to figure out exactly how can we amplify seed for species that are very, very difficult to make large collections from in the wild. And so you might ask, well, how do I know whether species on my target list are able to be stored, whether they're exceptional in some way? And unfortunately, there is no just like absolutely comprehensive resource that will tell you that information because so much more research really is needed. But something that is a great resource is the seed information database that's hosted on the Society for Ecological Restoration website. So you can search for your species on your target list, and sometimes there will be this really great information. Here's a little module on seed storage behavior, right? So this says, well, it says seed storage behavior uncertain, but then under conditions, it says seeds lose viability rapidly in storage, even at low temperatures. And it also says seeds require 3 months dry storage to overcome dormancy, right? So that's really good information to have. So it might be worth, if you've created your own target list for restoration projects in your area, to sort of run those species through the database and see if there's any information that you can pull out that will be helpful, useful to you. There are other factors that impact seed longevity, and one really important one that I want to talk about is the maturity of your seed at collection. So, it might be tempting, right? When you go out in the wild, let's say, you're out there making a seed collection, you have your whole field schedule planned out and it's pretty busy and you drove all the way into this population because the seeds should be ready, but they're just slightly green. It might be tempting to just go ahead and make a collection because you're like, oh, I don't know if I'm going to make it back out here. But one really important piece of information to know is that sometimes it's that last little bit of maturation that makes it possible for seeds to be long-lived. Like that's what they need in order to be storable, essentially. And so this is a graph that I took from this 2017 publication looking at soybean seed longevity at different maturation stages. So they collected soybeans at these different stages of maturity, and then they ran these accelerated aging experiments. So accelerated aging is just where they expose seeds to high humidity and high temperature, and it essentially makes them rapidly age. And the rate at which they rapidly age is supposed to be proportional to how they would do at ideal conditions. And so from this graph, we can see that the seeds that were the most mature lived the longest. So and the sort of the reason for that is kind of the same reason why some species might be recalcitrant, is that it's sort of like those last stages of maturity is where the cells are accumulating dry mass, and that dry mass is what they need in order to be desiccation tolerant, so we can dry them so we can store them. So sometimes, right, it might seem like some species appear to ripen up post-harvest, and you can collect slightly green and they ripen. But a lot of species, those seeds, if they're collected green, will not live as long, right? And we want our collections to live as long as possible so that they can be used in the future, right? We don't want them to be put into storage and then die right away because that's sort of a waste, right? We want our seeds to be able to be effectively used for restoration, for reintroduction, for research. Another thing that can impact seed longevity is the treatment of the seeds during seed cleaning. So this might not apply to everyone. Some collecting teams are just sending their seeds off to a seed processing facility and they don't participate in seed cleaning themselves. But for those of us who do participate in seed processing, just note that you can damage seeds through seed processing. And sometimes if you damage those seeds, they might not be able to be stored. And so I usually, when I'm processing seeds at CalBG, I'll take a small sample of my collection and process it and then take a look at the seeds to make sure I'm not damaging them before I process the whole batch. And then the last one is a big one, is humidity and temperature. So what are the actual conditions we're storing the seeds at? So the key things we need to keep seeds alive long term are low and constant humidity and low and constant temperature. And I sort of alluded to this in some of those previous graphs, right, where storage treatment is, having this big effect on seed longevity. And here's another one. So this is a collection of Ericameria nauseosa. They took a single collection and split it in two. So half of the seeds they processed, dried, and stored in our freezers like we normally would. So that's the blue line. And then the other half they processed, but just kept on somebody's desk in the office for a couple years and ran follow-up tests. And after just a single year at ambient conditions, a majority of the seeds in ambient conditions died. After 3 years, all the seeds were dead. Compared to the seeds that we dried and got into cold storage, they stayed alive. I mean, we ran a follow-up test in 2021, and the seeds are just as viable as the day they were collected in 1996. So we know that these methods work, right? These seed conservation methods where we're drying collections and storing them in the cold work for keeping seeds alive. And it's just sort of a fact that seeds cannot survive as long in ambient conditions. I would say that not all species are quite as sensitive as this Ericameria, right? Some species certainly can live in ambient conditions much longer, but because we don't know all of this information about which species are hardier, have hardier seeds, and what sorts of conditions different species can survive in, it's best practice to just treat them all as if they are this sensitive, as sensitive as the Ericameria nauseosa, and trying to get them sort of processed and into storage, and keeping humidity and temperature low, to try to prevent as much seed death as possible. There's sort of these Harrington thumb rules that for each 1% decrease in moisture content, the storage life of the seeds is supposed to be doubled. And same goes for every 10-degree Fahrenheit decrease in temperature. So these things really greatly affect how long seeds can survive in storage. There's also this graph. This whole slide was actually in another presentation I gave about post-collection handling of seeds, but I'm bringing it back because I like it. So this is screenshot of a technical information sheet that Q Millennium Seed Bank puts out. They have this series of technical information sheets that are really handy and have a lot of useful information. But I really like this one that shows, so there's equilibrium relative humidity. So the humidity of the actual seeds at these different development stages, right? So first the seeds are being formed, right? Reserve accumulation, right? So they're getting all those, the cellular dry mass. So they're maturing up. Right, there's an optimum time to collect, right? Got to wait until the seeds are absolutely ripe to go ahead and collect them. And they're sort of drying down through this process. And then this whole section is after harvest, what is the humidity of those seeds, right? And that's going to be fluctuating, right? With ambient conditions, there's fluctuations in relative humidity overnight, right? Temperatures drop and humidity rises. And what this graph is saying is that, if your seeds are getting above like 85% relative humidity, there's a really high risk of mold. And mold kills seeds, right? So your seeds are at high risk of dying if they get up into this category. But even here, between 50 and 85% relative humidity, that is also too humid, and your seeds have a really high risk of aging. So your seeds are going to be dying under those conditions. So the good rule of thumb is that we want to try to keep seed collections under 50% relative humidity after we collect them, to try to keep as many of them alive as possible so that they can be used effectively for restoration. And then there's a lot more information about drying seeds and options for how to dry them in that other YouTube video on post-collection care of seeds that you can find on the Garden's YouTube channel if you want to check that out. So from this sort of first section, some tips for seed collectors. First, research target taxa to determine whether they might be exceptional species before attempting a collection. 2, only collect fully ripe seeds, even if it seems like they ripen post-harvest. And 3, keep seed collections cool and dry and process and store them as soon as possible. And then this is a quote by Dr. Chris Walters as one of my favorite quotes. Seeds are not rocks, right? Seeds are alive and they are dying, and we're trying to treat them as if they are living things that have the potential to die, right? And trying to be nice to them to prevent as much death as possible before we need to use them. Okay, so moving on from seed longevity, right? So it's really important that we are collecting seeds that are able to be long-lived so that those collections can be used effectively in the future. But it's also important to collect viable seeds in the first place, right? That are viable upon collection. So not just long-lived, but healthy, right? Or have initial health. So viable seed has the ability to germinate and grow into a healthy seedling. And most often, some portion of the seeds that you collect will not be viable, right? A plant population is going to produce a lot more seeds than what will actually result in plants, right? And the reason that we want to talk about this is that usually restoration seed collections have some sort of goal for quantity, right? So for seeds of success, a standard collection is 10,000 seeds and an operational collection is 80,000 seeds. But it's not just 80,000 seeds, it's 80,000 PLS, pure live seed. So you actually want 80,000 viable seeds. And so we can sort of do, right, before we make a collection, we can start by doing just a visual assessment of the seeds. So actually taking a sample of seeds, trying to get them out of their fruits and taking a look at them and seeing, do these look healthy? And this might seem like obvious advice, but people definitely skip this step. Usually not, right, SOS collectors, because it's part of the SOS protocol that we have to do this. But people definitely skip the looking at the seeds part. And I know this because I literally just got a collection deposited at the seed bank recently of Areodictyon trichocalyx, where all they collected was galls. So these are galls. They look like fruits, but this insect, is affecting the plants, and these galls will form on the inflorescences. And if you're just looking at them, right, they're looking like fruits. And so somebody thought, oh, that must be the fruit, and that's what they collected. But all they collected was galls. So they actually made a collection of like 100 individuals, and there were only 300 seeds in the collection because they didn't collect seed. So that would not have happened, right? If they had actually like taken a look and looked for healthy seed before starting a collection, they could have saved a lot of time. Sometimes, right, if you're taking a look at your seed, you can sort of tell which ones seem viable based on their size or their shape, right? In this Lysyngia, the seed on top is like a little bit more plump. That's the filled seed, and the seed on the bottom is sterile. There's nothing in there. Sometimes you can also do like a pinch test where you can feel whether they're filled or not, right? If you pinched the seed on the top, you would probably feel something solid in there, versus the seed on the bottom, if you pinched it, it would just completely crush in your fingers. So that is sort of one way to tell. Sometimes you can tell by the color of the seeds that healthy seeds will look darker than non-healthy seeds. So this example of Salvia mellifera, the seeds on the left are the viable seeds and the seeds on the right are the non-viable seeds. Sometimes you can tell whether seeds are healthy based on whether there's proof of, insect predation. There's little holes, entry or exit holes in these seeds. Yeah, those seeds are not healthy. Those seeds have been eaten. If you cut those open, you would find a little bug instead of an embryo. But sometimes seeds can look totally healthy on the outside, right? They can be the right color, shape, size. They won't have little entry holes or, obvious signs of predation, and that you won't even really be able to do the pinch test, but they still will be unfilled or unhealthy. And so that is why we do cut tests, right? Cut tests are very important. This is a standard part of the SOS protocol that we all need to be following. But, even those of you out there who aren't on SOS teams, just so you know, it's a good idea to actually take some equipment out so you can cut open a sample of your seeds before you start a collection to make sure that you have healthy looking embryos in the collection. So you can take a sample of seeds, cut them open, and a healthy seed should be pretty much filled with an embryo. It should be sort of whitish and moist. So if you cut open a seed and it's empty, or it has a, shriveled little gray embryo, right? Those are not living seeds. And when I say a sample, this is going to vary a bit depending on your team, right? Like your supervisor might have specific information for how they want you to do this. I looked it up in the SOS technical protocol, and I don't think it's written like a specific number to cut open or a specific number of plants to sample from. So definitely, check with your supervisor. It might be in the digital data collection. I haven't checked there. But in general, right, you don't want to just sample from one individual, like a small number of seeds from a single individual, because that might not be sort of representative of the population, right? You at least want to sample from some sort of random set of individuals in the population. And so in this example, we chose 5 individuals randomly in the population and cut 10 seeds open per plant to get an idea of how filled the seeds are. Or you could make a small collection from a larger number of plants and then combine those and then cut those open, a sample of those open. And it's just a really important thing to do, just to make sure that the seeds at least appear to have healthy looking embryos before starting a collection. Because it's actually fairly common for plants in the wild to just not fill their seeds. Like they might flower just fine, they might start to produce seeds, and then conditions are just not right. They're just going to reserve their resources and they don't actually fill their seeds. And so it's probably good to know that before you start a collection and collect all this material and spend all this time on it, just to bring back unhealthy seeds. So for actually doing these cut tests in the wild, this is some of the equipment that you'll need. So some seeds are large enough where you can just cut them open with nail clippers and just use your eyes to see if they look like they have healthy embryos. Those are my favorites. I love those seeds. Nice big seeds. But for all other seeds, right, you're going to need the rest of this equipment. So we usually bring a clipboard. It doesn't need to be a clipboard, but something hard and flat that you can use as your cutting surface. We'll bring double-sided tape. And so we put double-sided tape on the clipboard and then put our sample of seeds on the tape so it sticks down. And then with your loop and your blade, you can cut them open and see what the embryos look like. Sometimes you also need tweezers so you can actually hold seeds down if they're jumping off of your double-sided tape. But this should work even for small seeds. So I just did this the other day with sedum, which have very, very tiny seeds. And just with the loop and that little setup, I can see embryos. Sometimes you can also kind of feel, like if you go to cut open a seed and it just crumbles under you, you know that seed wasn't filled, versus like when you cut open a healthy seed, it's got some texture to it. Some species might be very difficult, though, to do cut tests on, like some grasses just don't really seem to have like meaty embryos, I guess. I think they're just sort of like long and thin and flimsy, so those can be a little bit tricky to determine. And so if you are coming across that, just check with your supervisor and see what they want you to do. If they want you to go ahead and make a collection, make a partial collection, bring it back and try to do cut tests under a microscope in the office, or just make a collection and sort of see what the results wind up being. And that will inform that for future collecting team for that species, and what kinds of numbers you can get. Okay, so seeds, even though seeds might have healthy looking embryos, but that doesn't actually mean that those are living embryos. That just because an embryo looks healthy doesn't mean it is actually viable. And so, right, because viability is the ability of a seed to germinate and produce a healthy seedling. And so there's also, on top of that, like after the fact, we do viability testing on seed lots for storage, right? So at CalBG, we do germination testing on all of our collections that are coming in. to get, baseline viability information, but then we also do follow-up tests through the storage term. So we're monitoring how our seeds are holding up and gaining information about how long-lived seeds of California native plants are. And the way that we do germination tests, we just plate a random sample of seeds on agar. So agar is just a nice moist substrate for germinating seeds, and it's clear, so you can see when the radicals pierce the seed coat and count it as a germination. And we'll just see what portion of those seeds germinate, right? And that gives us an idea of how viable those seeds are. Some people will also do tetrazolium testing. This might be something that you've heard of, where it's a biochemical test. Sometimes people will do both. They'll do a germination test, and then on the ungerminated seeds, they'll do tetrazolium testing, or sometimes they'll just do tetrazolium. But it's a biochemical test where seeds are soaked in tetrazolium salt solution. And if the seeds are respiring, right, if they're living, respiring seeds, then this red compound is formed and the seeds are stained red. So in this image, the seeds on the top they have been stained red. Those are living seeds. The seeds on the bottom are not viable. And I guess the main reason I want to bring this up, this idea of viability testing, is just to note that even if, like, if you're trying to meet some target, like 80,000 seed collection, but you want it to be 80,000 pure live seeds, just note that even if you do your visual assessments and you do your cut tests and you're using that to estimate how much you need to collect to meet your goal, you probably want to collect a little bit more than that, because some portion of the seeds, even the ones that look healthy, won't be viable. Yeah. And so one last tip to mention is to collect this season's seeds, because sometimes plants can hold on to old fruits, hold on to old seeds, even sometimes for years. And sometimes those seeds visually they look healthy, but they might be much less viable, right? Because they've been sitting out there in ambient conditions on the plants, for at least a year. And so this is a graph where they made a collection of Gimbelia speciosa from the Channel Islands, I think in 1996. And they made collections from current season fruits and previous season fruits, and then compared their germination rates and found that the current season fruits were much more viable. Sometimes when you're in the field, it can be a little bit tricky to determine, well, what's the current season fruits versus the previous season. Sometimes you can tell just by, like, the previous season fruits will look like they've been out there for a year, right? They might have like cobwebs on them or be discolored. But also, right, this is another reason why scouting and phenology checks are really important, because you can go out while plants are in flower and then sort of track the phenology of those flowers and sort of see them produce fruits so that you know that they're current season. And so some tips for seed collectors based on this last section, right? Always conduct your cut tests in the field before starting a seed collection. Collect extra seed to account for non-viable seed in order to reach your PLS goal, and also collect this season seeds. And that is actually all that I have for you guys. So we have a little bit of time, and I can take questions.
Yes, so we have quite a few questions that have come in, as well as a heart and some other things. So very nice job, Cheryl. So I'm just going to kind of go through what's come in, and you can answer or direct us to resources. So the
Great.
first question that I have for you is if you could please describe your storage techniques and methods.
Yeah, how we store our collections.
Mhm.
So we dry our seed, so we process our seeds to try to remove as much excess plant material as possible. So the storage techniques that I use at CalBG are primarily for long-term conservation. We're trying to keep seeds alive for like hundreds of years as like a backup to the wild populations. So we try to actually remove as much excess plant material as possible. We try to just store pure seed. So we do a lot more seed processing than like a short-term collection that's going to be used in the next 10 years for restoration. And then with the way that we dry our collections, we just have them in like terrariums, but like an enclosed container with silica. And so the silica absorbs the moisture from the seeds, and I'm able to measure the humidity of my collections. I have a hygrometer for making sure they get within this range that's ideal for humidity. And then we store them in sealed containers in chest freezers. And so it's really, it's not a whole lot of fancy equipment, right? Like my chest freezers I just got from like Home Depot or Lowe's, they're just home grade chest freezers. My drying technique is very low tech, just those terrariums. I made them myself. But doing those steps really has the potential to keep seeds alive for a very, very, very long time. For larger, like restoration size collections, a lot of times, you don't really have freezer space. You don't necessarily need to be freezing them, and just cool, dry storage is fine. So you can, dry seeds down in a larger, drying chamber and then keep them in like refrigerator storage.
All right, great. This question is related, but from a different person, but it's just, I think, sort of a clarification of what you just asked, so I'll ask it anyway. So should you allow most seeds to dry a while in ambient conditions prior to cold storage, or go straight into cold storage?
Yeah, you should definitely let them dry. So you can dry them in ambient conditions, and then store them in a refrigerator. That is fine. But if you want to freeze your seeds, you actually need to do more than ambient drying. You need to dry like over silica or like sodium salt solution or something, so that you can actually get them to this particular range of relative humidity. Because if you freeze seeds that have too much moisture, even in like the cells of the seed, like when water freezes, it expands, right? So if there's too much moisture in the cells of the seed, it can freeze and expand and break down cell structures and actually do more harm than good. So when we're preparing seeds for freezer storage, we have to do more than just ambient drying. But refrigerator storage, usually you can get away with ambient drying, or you can still use silica. Honestly, drying seeds is like the most important thing for keeping them alive. So anything that you can do to help your seeds get nice and dry is the best.
Excellent. Another question for you. So what are some strategies for preserving those exceptional plants that don't have seeds that are suitable for long-term storage? Is it just a
Oh,
matter of
yes.
continuing to have live specimens and collecting the seeds for immediate use?
Such a good question. Yes, so there are other methods of ex situ conservation for those exceptional species. Sometimes, right, we can use cryopreservation. We can like cryogenically freeze just embryos or just like apical stems. Like people do all kinds of crazy stuff. There's micropropagation. We don't have any of those facilities, and I don't have very much experience with those types of methods at CalBG. But one thing that we do participate in is conservation of species through our living collection. So, we have an 86-acre garden. We have of all California native plants, and those collections are accessioned. We know where each of those plants was collected. We have information about that. But we also have like what we call conservation groves at the garden, where it's not just like a single representative specimen of a species or a couple individuals. We actually have like a backup population growing at the garden, where each individual at the garden was collected from a different parent in the wild, and we have like backup populations, and then we also send backup propagules to other institutions. So there's a backup of the backup. So there, yeah, there are other methods for conserving species that are exceptional, and probably right the most important way to conserve species that are exceptional is through in situ conservation, right? To conserve them where they are in the wild. But in situ conservation is very much lended by ex situ conservation, conserving them outside. So just in case something happens that's unexpected in the wild, we have some sort of backup to fall on.
Great. Okay, another question. Can you provide links to the papers that you've cited?
Yes, I know. I wanted to put, it occurred to me that I did not put like a citation list on this slide, and it was too late to add it. So I can definitely do that and actually have a list of all, how can we send that to the participant list, Jennifer?
That is a great question. I think I can send an email out to everybody
Okay.
over Zoom. If for some reason we can't, and you particularly want one, like if I haven't provided you with some way of doing that in the next couple of days, feel free to send us back an email and we'll make sure that you get that. But I think Zoom will let me actually send something out to everyone.
Yeah, I also should have put my email address on this slide, and I didn't do that either. But my email address is on the garden's website, so you can easily reach me and also ask any follow-up questions that you don't think of during this session.
All right, perfect. Let's keep going then. We've got lots of questions. So another question, is there research to suggest that we can identify trends of viability within a single genus? So for example, if we know the viable storage life for Rubus or Sinus, can we infer that other species in that genus also have similar storage life?
Um, so yes and no. Sometimes there's definite trends within a genus for how long-lived things are, but sometimes there isn't, right? Sometimes it actually might have to do with like what habitat those species grow in. Some areas of the world or certain habitat types there's a higher proportion of seeds that are short-lived for whatever reason. There's a lot of research that goes into seed longevity and trying to predict seed longevity and look for trends in seed longevity. It's this whole field of research that's super interesting. And so, yeah, sometimes you can sort of sometimes you can estimate how long seeds will live based on similar taxa, related taxa, but not always.
Great. All right. Next question. So at the CalVG seed bank, do you store any seeds in refrigerated storage or do all the seeds go into freezer storage? And if you do store within refrigerator storage, what factors do you take into account if it should be in one place or the other?
Yeah, so in the California seed bank, which we're trying to conserve species long term for conservation purposes, we pretty much have everything in the freezers. But also at CalBG, we do have a restoration program, and we make restoration collections. Like for our national forests and for certain restoration projects. And so we also have refrigerator storage at the garden. So sometimes it's more a factor of what are the seeds going to be used for and when will they be needed in order to determine whether they go into refrigerator or freezer storage. I have a couple things that I have in the refrigerator that are sort of experiments. I have lots of little experiments going on, and sometimes we'll experiment with trying to see how long can this thing live just in refrigerator storage. But in general, we don't keep our like permanent accessions stored in the freezers. I mean, in the refrigerator.
All right, great. You ready for the next one?
Yep.
All right. So, have we at CalBG contributed to the propagation protocol database that's currently being developed by the state Native Plant Society, the California Native Plant Society? It says there's protocols for roughly 300 species, both seed and vegetative.
That is a really good question. Have we? We might have. I'm not sure. I know that I also share our germination data. So just in case anybody is interested, on our garden website, you on like the seed conservation page, I have a link to a summary of our raw germination data. We have like 40 years of germination data that is sort of hidden away in our database. And I just publish a summary of it. So it's just like a single row per test. So there's a lot more information than is just what is on this spreadsheet. But I shared that. I want to make it public so that it's not just hidden away and people can use it. And I've had various people reach out and ask if they can use it for different sort of compilations of data like that. And I can't remember whether this propagation database was going to use any of our data, but I'm very free with my data. I'm not trying to hide it away. But we also have, a whole restoration nursery, production nursery at the garden. I'm not sure if any of the staff over there have contributed to this specific resource.
Okay, great. All right, next question. Does GA3 break dormancy, or is it strictly used to hasten germination?
Oh, what a specific question. Yeah, so GA3 is gibberellic acid, so it's a growth hormone. And when we are doing our germination tests, so one of the reasons we do germination testing is to try to figure out how to break dormancy and get seeds to grow. And usually what we're trying to do is like expose the seeds to what they might experience in the wild and try to figure out how to break their dormancy. But one thing that you can do that's, I consider it slightly cheating, is you can just soak them in a growth hormone and get them to germinate. So sort of a way around figuring out how to break their dormancy. And so, yes, it breaks dormancy, it gets the seeds to germinate. It is a little bit tricky to use because you need to, like, different species want like different protocol to be used, right? A different concentration or a different soak time. Sometimes if you use too high of a concentration of gibberellic acid, you can harm the seeds. And if you don't use a high enough concentration, you can, like, nothing happens. And if you use just a little bit too high of a concentration, you can get the seeds to grow, but they're all leggy and weird and they die. But we definitely use gibberellic acid at CalBG, both in our like nursery for propagation for things that we know to be tricky and absolutely benefit from gibberellic acid. And it is a way to get them to produce, to get them to germinate.
Super. Okay. Another question. Are there any downsides to testing seed viability with tetrazolium?
Are there any downsides? Okay. Well, so I don't, let's go back to the tetrazolium slide. So we don't actually do tetrazolium testing at CalBG. There's definitely like positives and negatives of tetrazolium testing, compared to germination testing. So with tetrazolium testing, it's actually like very quick. So with germination testing, when we run a germination test, sometimes they last just a couple weeks. Sometimes they last a year. Like we just keep the seeds in the agar for a very long time. And so each individual test can take a long time. With tetrazolium testing, you can use, a smaller sample size. You're like usually excising the seeds or nicking them in some way and soaking them like overnight. The whole test is usually done within, 48 hours. And so that is a great positive. One downside of tetrazolium testing, so it is, different species have different protocol. And so if you're just working with like a small number of species and you can sort of like perfect how to conduct your tetrazolium tests, like what concentration to use and what soak time, and your seeds still need to be pre-treated usually to break dormancy in order for the test to work. So you still need to be, treating them. And then also how you analyze the results is different depending on the species. So it's like a very specialized thing. So for some species, see how like this little part is red? Some species, this might be a positive test, and others, it's a negative test. Some species, this might be a negative test, and others, it's a positive test. And so because it's so species specific and like the specific protocol needs to be developed per species, that's why we don't use it at CalBG because we're working with so many different species and we don't have like the time and resources to really dedicate to figuring out the protocol for each taxon. But there are a lot of reasons why tetrazoleum testing is really great and an absolutely valuable resource. Also, by the way, it's destructive to the seeds, so then you're staining those seeds and then you can't do anything with them. Versus germination testing, right? Just as a byproduct, I wind up with little babies, and for some of those babies, we could transplant to soil and then grow those plants up.
Super. All right. So what humidity do you store seeds at?
Okay, so we dry our seeds to like 25 to 35% relative humidity at room temperature, right? So relative humidity is relative to the temperature. So if we're drying at room temperature storage or room temperature, we dry between 25 and 35%, seal them and put them in the freezers. And so the humidity actually decreases of that collection. In freezer storage, it's more like 15 to 20% relative humidity in the freezers.
All right. And then do you have any specific protocols for pulling seeds out of storage and rehydrating them for viability testing or for propagation?
Yeah, the main thing is that we have to let our seeds sit and come up to temp before we open the storage package. So if you just take something directly out of the freezers and open it, the seeds are going to absorb moisture and it's going to damage the seeds. So we pull them out and we let them defrost before opening them. But then they're pretty much ready to go for propagation or for germination testing. The main thing is to try to figure out like what pre-treatments they might need to break dormancy, which vary depending on the taxon. And so some things need a water soak, some things need a cold stratification or to be soaked in smoke water or boiling water or what have you.
Okay, great. Next question. How economical, affordable are the germination TZ tests? Does it require specialized training and equipment? Or is there anything they should know?
Yes, it definitely requires specialized training. I have this big manual. I'm pretty sure it's available for free though. Maybe not. You might have to buy it. I have this big manual on doing tetrazoleum testing. Um, I'm actually not sure because I haven't actually done tetrazoleum testing myself. I'm actually not sure how expensive the equipment is or like the tetrazoleum salt solution. I imagine it's not terribly expensive.
Okay, thanks, Cheryl. Another question. So this comes from somebody who helps to manage a native seed library, so like a free little library, but with native seeds to
Mhm.
encourage folks to plant natives in their garden. And they say that they bought locally collected seeds in the past, but they're trying to sustainably harvest seeds from their nature reserve properties to fill the library. And so
Mhm.
they want to know how long would keeping dried processed seeds in jars with silica at room temperature, like how long would they store? And then when they pack the seeds into coin envelopes to go into the seed library, how long do you think those might last?
Oh my gosh. Well, the answer is, I swear, I'm gonna get this tattooed on me so I can just like hold it up every time the answer is. It's species specific. And I feel like this is the answer to everything. Some species are going to be quite long-lived under those conditions, and others might die pretty quick if they're just in the coin envelopes. Like particularly, depending on what area you're from, I feel like I might know who this person is. And if you're in the Los Angeles area, right, it gets hot, right? It gets pretty hot out here. Keeping seeds in those little library things, just in ambient outdoor, where the sun is beating down on them. Some species are definitely going to die under those conditions. And so, yeah, it might just be a factor of trying to keep them in the jars as long as possible and then just stocking a small amount into those free libraries. But honestly, the same goes for, keeping seeds. If you have them seeds in jars over silica, keeping them dry is even more important than keeping them cold. But if you can stick that jar in the refrigerator, that will definitely extend the life of your seeds. So if you just put it in the fridge and you're good to go.
All right, great. Another question. So, is there a database where we can see what species are like an orthodox species, an intermediate, or a recalcitrant species? Is there a list?
No, there really isn't. The best resource probably is the seed information database, because many species do have, at least the ones where we know that they are recalcitrant or that they are intermediate, will have that information listed in that seed information database, the Society for Ecological Restoration. Um, but a lot of stuff we still don't know, and there's so many different species. Especially, I mean, California is so botanically diverse. A lot of these things nobody's ever tried to seed bank them before, and so we're still learning that information. Yeah, I wish there was though. I wish there was just like we talk about it sometimes, like establishing a resource like that, but it would still we would be very slowly adding to it as we learn more.
Okay, great. I have a couple of bug questions coming up next.
Oh, okay.
Do you have any tips on removing bugs from collections?
I do. Okay, so usually when I am just like processing seeds for my own collection, and I notice a bug issue, usually what I do is prioritize that species for processing, because usually just processing, you can get a lot of the bugs out. And then because I am storing in freezers, I can just sort of prioritize that collection for drying and freezer storage, and that kills everything, right? I don't have to worry about bugs in my frozen collections. But for things that are going to be in refrigerator, dry storage, um, what I would recommend, and this is on the Seeds of Success technical protocol, which is available online for free if you go on the BLM Seeds of Success website. There's actually two different protocol that they use, and I recommend the one that NPS, National Park Service, uses, which is to use a product called Taro Garbage Guard, and you put that in a trash can with your seeds for 72 hours. And there's like specific protocol for how to make sure that you're doing this safely, following the instructions for handling pesticides, right? But that's the protocol that we're actually switching to at the garden, is using the Taro Garbage Guard for killing pests, seed pests.
Okay, so that is related to, I think, my next question, which is about using insecticides after wild collections. Is there any damage that comes from that? I'm assuming that the Taro Garbage Guard is an insecticide. Okay.
Yes, yes, it is. No, I don't think that the seeds are damaged by using them. Just people, just be careful. It's a pesticide, it's harmful to people.
All
Safe
right,
handling.
great. And you answered a question about freezing to kill bugs also, so thank you.
Yeah,
All
yeah.
right.
Just don't freeze them if they haven't been dried. Because I get that question. People are like, "Oh, I'll just come back and freeze my collection right off the bat to kill everything." But you're probably going to kill the seeds too because they haven't been dried to the right level for freezer storage.
All right, good additional detail. Actually very related to that. How long do you dry down your seeds, or does it depend on species?
Uh-huh, yeah, it's species specific. Okay, so we used to, before we got our hygrometer, we used to just put our seeds in those drying tanks with the silica for 2 weeks. We just had a standard set of time that they stayed in there. But then we got our hygrometer, and now we can actually measure what's going on with the seed samples themselves and see what humidity they are. And what we have learned is that it's very species specific. So some things can just be put in the dryer for 24 hours and then they are ready. Other things need to stay in there for more like a month or 6 weeks or something until they're dry enough. So it really depends. But we got by for a long time with our 2 week time frame, and a lot of things seem to be ready in 2 weeks.
Very helpful. Some other questions about drying. So someone is asking, I'm assuming your terrarium jars are airtight. Would a mason jar be essentially the same?
Yeah, anything you can use anything airtight with some silica and it'll do the trick.
All right, great. And then just make sure I'm not missing anything. Okay, and then we have some questions about just some other resources. So would it be possible to share the presentation slides also?
Oh, that is a good question. And I feel like we had decided something about this during the last one. So the presentation itself is going to be posted on YouTube. I think that we wanted to not share the slides and just have the YouTube video available, where you can see the whole presentation just there.
Okay, perfect. And then sorry, just making sure I understand our questions. Are you planning, so somebody asked about, are we planning a seed germination workshop in the near future? So I can answer part of that and you can answer part of that.
Oh, are you planning one, Jennifer?
Yeah, so it's not related. Cheryl is not teaching that with us, but we are doing well, it's a seed propagation workshop. So,
Mm.
it's not germination specifically, but it is our nursery staff are going to be talking about, for growing in your home garden, how to do, how to get seeds to germinate and propagate that way. But I don't know if you have anything planned for like germination testing or anything like that.
Okay, so I don't have anything planned, but I can tell you of this really wonderful resource that is the Center for Plant Conservation has a applied plant conservation course that is free. It was funded by the Bureau of Land Management, and there's these series of modules and all these videos that are instructional videos. And I had put one of them together, I had put a couple of them together for this project, but I have videos on germination testing that are available through the CPC website for free through this applied conservation course. And one of them is even like step-by-step guide to how we do our germination testing. So I highly recommend checking that out. And then if you have any additional questions, like feel free to email me. I'm an open book. I'm really happy to share information.
Excellent. A few other questions about resources and also, so we had an ask for your raw germination data. That's on the website, correct?
Yeah, it's available on the website. There's like a button you push and it will download an Excel document.
Okay, perfect. And if I can send out the email, I'll send out a link to that as well. And then a question about sharing your germination testing protocols.
Hmm. Oh
Um,
yeah.
yeah, are you able to do that?
Yeah, I mean, same answer. You can find exactly how we do our germination tests on that Center for Plant Conservation Applied Conservation course. I have a video about it. It shows like the products that we use and like how we make our agar and everything. And also, I'm like more than happy to talk to people about what we do and to share specific protocols if you want to email me.
All right, great. And then I believe it's our last question, but kind of going back to the GA3, how do you know how much to use? Is it related to seed coat
Hmm.
thickness?
Yeah, how do
I
you
should have
know
catched.
how much to use? We have sort of a standard concentration that a lot of things we will do like 200 or 500 parts per million, but I've heard of people doing much higher and I've heard of people doing lower. So I think it's species specific. I don't think it's necessarily related to seed coat thickness or even seed size or anything. It's just different species have different preferences. And it takes a little bit of playing with to sort of figure it out for the taxon you're working with. But we for our germination testing, we'll frequently use 500 parts per million because like that might be too high for some things and produce weird seedlings that aren't able to survive. But the main purpose of our germination testing is just to see whether they're alive and able to germinate. And so we don't care as much about whether they can then survive. And so 500 isn't as risky for us, I guess.
Okay, thank you for that clarification, Cheryl. All right, I believe that is all the questions that we have right now. I think I got to everybody's. For some of those follow-up things, I am going to try and send out an email to everybody with some of those resources. So I'll talk with Cheryl and make sure I get a list of those to send out. As I said before, if for some reason I am not able to send out that email to everybody, feel free to reach back out to us through our website, and we can send you any links or any resources that you need. So thank you again for joining us today. We really appreciate having you all here and all of your insightful questions. And I hope you'll be able to join us again for some of our programming in the future.
Yes, thank you. Thank you so much, everyone. Thanks for coming to the talk, and just, yeah, definitely email me if you think of any other questions.
Wonderful, thank you. And we will be posting this onto our YouTube channel within the next couple days as soon as we get everything edited and ready to go.